Hi, good afternoon, everyone. Thank you for coming to the talks today to listen to some really insightful, learn more about cancer. My name is Wei Liang, and I'm a research fellow in Associate Professor Polly Chen's lab in CSI NUS. And today, I'm actually here to share with you our recent work on studying how, when your body fails to recognize itself, liver cells and immune cells can actually talk to one another, and this can control liver inflammation. So to maintain health, right, you need a balance between immune reactivity and immune tolerance. So immune reactivity is actually when your cells get infected by foreign pathogens, such as viruses. Your body senses this and can start inflammation to fight the virus. But on the other hand, it's also important for your body to be able to recognize itself as self and not attack healthy, normal cells. And that is self-tolerance. So when this balance is shifted, such as when you have too much immune reactivity, you will have excessive inflammation, and this can cause autoimmune diseases. But if you have too much self-tolerance, actually you can get immune evasion, where your body fails to recognize that there are cancer cells present and non-inflammation to attack them. So recently it was found that double-stranded RNA and RNA editing plays a very central role in this whole maintaining this balance. So RNA is actually a very essential and abundant molecule, copied off your master copy DNA. It gives rise to proteins that can then perform all of your life's biological functions. So one way to look at RNA is as a passport. So when you enter your home country, you take out your passport and you give it to the immigration officer, who then inspects it. And if it's okay, what they'll do is that they'll stamp it a pass using RNA editing. And this will actually license your RNA to go through security without causing any inflammation. So this is actually self-tolerance. But viruses such as COVID-19, which is actually an RNA viruses, also have their own viral RNA passports. But your immigration officer, when checking this, won't stamp it, and because it is not stamped, instead this will trigger your immune security to mount inflammation to kill the virus. And this is actually immune reactivity. So interestingly, it was recently found that cancer cells actually can exploit this passport stamp system. And how they do this is that cancer cells can have so many copies of this stamp and so much RNA editing. They actually can disguise the cancer RNA and hide the cancer cell from your immune security. Because in this way, no inflammation is mounted to kill the cancer cell, and they can continue to survive. So a lot of work has been done to understand how these cancer cells can actually exploit this system. But we still don't really know how your immune system reacts to this. So that is what we wanted to find out. And to do so, what we did is that we used mice, and we removed either the stamp or the cell's internal security to see what will happen during loss of self-tolerance. So it is known that if you have a loss of self-tolerance, you have no RNA editing, basically you have no stamps, these mice become very, very sick. And unfortunately, they can't even be born, because there's so much inflammation that their liver actually disintegrates. But if you remove the cell's internal security, you can see now that these mutant mice actually can be born, and they look quite like their normal siblings. But if you look at their livers, they still have liver damage and liver inflammation, even though the internal security is already removed. So this was the problem that we wanted to find out why this is happening. So what we found out is that during loss of self-tolerance, your liver cells actually can accumulate a lot of unstamped RNA, and this causes inflammation. And this results in sending signals being released, which are then picked up by active immune cells to call them to go into the liver to cause inflammation. And importantly, what we found was this inflammatory communication was actually reduced in liver cancer. So in liver cancer, you can see that liver cancers have very high amounts of RNA editing in pink. And because there's a lot of RNA editing, there are less sending signals being released by the cancer cells in orange. And if you have less sending signals being released, that also means that there are less active immune cells in green and red being brought into the tumor. So what this means is that in the liver cancer, this inflammatory communication is actually suppressed. And that ultimately results in reduced inflammation in the cancer and hence reduced cancer cell killing, allowing the cancer cell to continue to survive. So to summarize, what we found is that during loss of self-tolerance, a lot of unstamped RNA accumulates, causing inflammation and release of sending signals that actually call active immune cells into the liver to cause inflammation. But in the scenario where there are cancers that exploit this stem system and they have so many stems and so much RNA editing, it actually shuts down this communication. And your immune cells are not alerted to the presence of the cancer cell. So they're not brought into the tumor to cause inflammation to kill the cancer. So what this highlights really is that we found a very unique way where cancer cells can actually use this RNA editing to disguise themselves and hide themselves from your immune security. And we find that this actually presents a lot of different avenues for potential targeting for therapeutics in future. So I'd like to thank the Diana Koh Fund and Ms. Janet Lim, as well as my supervisor, Associate Professor Polly Chen, and members of PLC Lab, as well as the collaborators and clinicians that helped make this work possible. Thank you.
Okay,